But yeah, hello everyone, and thanks very much for joining us tonight. And we are very excited to have Dr. Natasha Rhys here to present to us. She's currently a biophysicist at King's College London, and her research is focused on understanding the structure and properties of biological, material, and drug molecules in solution. Natasha obtained her BSc in physics at Imperial College London, and then her master's in structural biology and biophysics at the University of Manchester. In 2011, she started her PhD at the group of Professor Lorna Jagan at the University of Leeds. She then worked as a postdoc researcher in the group of Dr. Sylvia MacLean from 2015 at the University of Oxford, investigating the solution structure of an increasing range of biological and pharmaceutical systems before moving to work with Professor Chris Lorenz in 2018. And in 2019, she was awarded a King's Prize Fellowship to develop her own research program. And now I'm going to hand over to Dr. Natasha Rhys to present her impressive research.
Thank you ever so much. So I'm just going to close this down. So basically, yeah, Tina's given me quite a lovely biography introduction there, so thank you for that. But yes, so as Tina said, I'm Natasha Rhys. I am a King's Prize Fellow. This is a senior postdoctoral research fellow over at King's College London in the Department of Physics. So basically, I feel like I'm about to go and do a talk that's going to summarize almost like the past 11 years of my research career. I guess there are some researchers who sort of work on a specific sort of scientific area, and they apply a lot of techniques to understand this. And I tend to be a bit the opposite, where I use a narrow set of techniques to see how we can apply this to study a range of different biological systems. And the aim of this talk is I'm going to summarize effectively how we might do this, what goes on, the approaches that we need, and what kind of questions we can answer. And as evidenced by my title, that's basically focused around, as it says, revealing the atomic level structure of biological molecules in solution. Okay. So let's go and set the scene, let's say. Let me move on. Aha, there we go. So when we think of biological molecules in nature, in biology, we can think of, say, the proteins, the nucleic acids, the DNA, the sugars, and the fats and lipids that make up our natural world and the molecules in our body. The one thing we have to remember, of course, is that natural systems and ourselves are made up of a lot of water. We are ourselves about 2/3 water, and this basically is what surrounds the molecules and tissues in our body. Therefore, ideally, if we want to understand the structure of these molecules and their properties, it's more ideal if we can study them in a phase akin to that in nature. So say where there's going to be water present. Now, biological molecules have a lot of advantageous properties to them. And if we can understand them, we can exploit these molecules to form a range of different biological materials. This is this ranges from things like nanostructures for material applications, for developing new therapeutics, possibly osmotes as well, a range of different structures of all these molecules. Now, even when we are forming these systems, the chances are we're forming them with some kind of solvent present as well. So what generally determines how biological molecules can assemble to form these different structures is based on two factors. The actual structure, the atomic level structure of the molecules that you're actually working with, as well as the solvent environment and the components in the environment that the biological molecules interact with. Therefore, once again, if we want to be able to understand fully the structure and the properties of these molecules, not just for understanding function, but how we can apply them to look at, say, material systems, we need to have an understanding of the role of both the biological molecule and the solvent on these assembly processes. And what's fascinating is the fact that we currently don't have very much information on the atomic level interactions that drive these processes. So, how is it that then we currently study biological structure in the first place? So over decades, and I tend to say a century now, we have made leaps and bounds in understanding chemical and later biological structures using crystallography, and say we have a lot to thank for it. Now, the caveat, of course, of crystallography, we can look at atomic level structure, but the problem is it's a solid state method. And so we can't fully get the appreciation of the impact of the solvent on these structures. We can turn over to methods such as small angle scattering. As I say, this is a technique you can do in solution phase, but it focuses more on bulk structure. So bulk complexes, such as the protein complexes that are being shown in this image, and not gain that atomic level resolution. We could go to something like computer simulation, and this is a very up-to-date modern way to do this and has, again, a lot of potential doing this, and you can gain that atomic level information of the structure in solution. Of course, the thing to note is the fact it's only based on computation and lacking maybe the experimental measurement unless you bring an extra method. And finally, we can go to things like spectroscopy, which you can get that sort of some structural information from, but it tends to be measured indirectly through taking other parameters. Therefore, if we want to gain information on the direct atomic level structure of a biological system in solution and include an experimental probe at the same time, what we have to do is we have to either do lots of experiments and different systems to cover all of these factors, or we have to find techniques that can actually tick all three boxes. So if it's going to be such hard work, probably one of the questions now to ask is, why should I actually bother in the first place looking at the atomic level structure of solution state? And the way I'm going to answer this question is actually by playing with my title of what I actually mean when we say biomolecules in solution. So there'll be many people out there who might read this title, not necessarily you guys on here, but there could be some, who could read it as biomolecules just conveniently in solution. And what that does, if you take that analysis, is the fact that you focus just on the actual biological structure itself. So what it's made up of, how it's actually folded, and its conformation. But the truth is when we're saying biological molecules in solution, we mean the whole system. We mean the biomolecule and the solvent as well. And what this allows us to do, it allows us to get that information, say on the structure and conformation of the biomolecules, but we can then extract information as well on those atomic level bonding interactions that are being formed between the biomolecule and the solvent that we have been searching for. We can do that by directly probing the lengths, mapping the solvation. We can also look at things such as the bulk solvent structure to see whether or not the presence of our biomolecule is affecting the solution in general, and other factors such as, let's say, orientations. Even if you had something like, let's say, a ligand interaction coming in, interacting with the system as well, say something like drug binding studies, you could also ascertain the impact of those on the system too. Now, the technique that I focus on, and has been the main part of my research for the past 11 years, is using neutron diffraction. And what neutron diffraction allows you to do is directly measure the atomic level structure of a biological system in solution, and of course is a technique, an experimental probe of this. Now, one benefit of using neutrons is that they're actually sensitive to hydrogen atoms in the system, which is advantageous for biological molecules because they usually contain a lot of hydrogen. Now, we complete our experiments at the ISIS neutron facility in Rutherford Appleton, and the experiments we do will then allow us to get information down to angstrom level that we have. Now, this is a method that I guess became quite popular for its use in the study of water. But over the years, what's happened, it's being used to study increasing range of different structures, biological, chemical, and beyond, of increasing size and complexity. Now, what we've studied with neutrons, when it comes to sort of this biological, maybe biomaterial, or even pharmaceutical realm, we have an increasing repertoire of different things that have been studied. This ranges from studying maybe just simple salts that are relevant to biological systems, say understanding the assembly of lipids, understanding the hydration of sugars for relevance to the sweet taste, understanding how we can look at biomolecular folding. Now, I can't go through all of these, but of course you're free to ask me about them all, any of them. And of course, I'm actually going to touch on these or a couple of these during this process of this talk. Now, one thing as well as just using neutron diffraction, what we try and do in this system, because we have a bigger long-term aim of not just having this as a single technique in its own right, but also seeing in the long term how we can connect it into the repertoire of different biophysical techniques we have out there to study structure. So What I'm going to basically do now is I'm going to walk you through the process of doing a neutron diffraction experiment. And the first of all, as I said, that basically when you do this, you have to go to a large scale facility. So we have the ICIS neutron facility here, which is a huge complex of things. So you go, you take your samples, you stick them in the beamline, and wonderful magic happens. Now, basically, the process you have to produce your neutrons is you are accelerating charged particles. And once you've accelerated them in pulses, this is basically a pulse spallation source. So you accelerate pulse charged particles and then fire them at heavy metal targets. And that sets off an internal intranuclear cascade process that allows neutrons to be released that can be siphoned off to different instruments. Now, we have different instruments that can be used on different two-target stations to do solution-based studies, whether it's in bulk or at atomic level. And the instruments we have at ICIS for doing neutron diffraction are SANLs and Nimrods, so it's the yellow one and the red one in this case. Now, when we go and do our experiments, we don't just do one system or one sample, basically. So what I mean when I say this is that if we're looking at a specific biological molecule at a certain concentration, a certain setup, we will do more than one sample for this. And this is a process called isotopic substitution. And I'll explain why that we actually do this. So what we have, when we actually measure our data from neutron diffraction, we get our data in the form of the total structure factor. And this is basically a measure of all the pairwise interactions in our system. And this is just an example sort of data that we would get from a biological molecule in pure water. Now, what we can do is when we do our substitution is we can substitute some of the atoms for a different type. In this case, we can substitute hydrogen for, say, deuterium. And when we do this, what we now have is the data in this case, if we were to substitute just deuterium in water, what you can actually see now is that the signal between this line versus the original one looks different. What that means, if you go and take different isotopic substitution samples and analyze them together, by analyzing them together, you can identify if, for example, one line compared to this line because of this change, you know what parts of the data attribute to which atoms that you varied, pretty much this way. So what you can do is you can collect different isotopic substitutions. We don't just have to change the solvent, we can also change, say, hydrogens for deuterium in our biological molecule, for instance, and we build up that repertoire of different isotopic substitution samples for analyzing. So of course we go to ICIS, we do our neutron diffraction experiment, and we collect our different isotopic substitution samples and measure those. And again, we measure those as a measure of the total structure factor, F of Q, the sum of all pairwise interactions in our system. And we measure this as a factor of Q, which is effectively the inverse of real space. So effectively, the information in our atomic interactions are sort of at higher values, while our bulk structure is at low values of Q. Now, on any data we get, we correct it for any background scattering because we are interested in having elastic coherent scattering. It is a diffraction experiment. Now, if we were to try extract all the information on all the pairwise interactions from effectively lines, we'd have a really hard time trying to extract information on, say, the carbons, nitrogens in our biological molecules, because everything is sat in these lines. And the chances are in a lot of systems that the solvent is going to be the major component of these systems. So most of the signal that you see here is from the solvent. So what we do to try get over this process of trying to extract those pairwise interactions is use a specialized piece of software called Empirical Potential Structure Refinement, or EPSR. And basically what EPSR does is it aims to create a model of the system that fits the experimental data. So the first stage that we have to do is we have to create a model, like a starting model to work with. So we go to the literature and we find the Jones parameters and charges from the literature and create our molecules to create a box of the correct molecular ratio and density. Now, when I say that we are getting potentials, starting potentials, what I mean is that we have to consider both possibly a starting conformation. So we might go get a structure from something like crystallography that's available to this, that has maybe good starting bond angles, lengths, torsion angles, conformation general that we could start with. Of course, this will not be the most optimal one, but we're giving it a starting structure to work with. And of course, we have things such as other force fields, such as considering Leonard-Jones parameters and charges that might be applied to the atoms in our system. And we have molecular force fields out there we can start with to try putting these in. So we have things like amber, charm, OPLS, and for things like water, we have SBC and TIP. Now, actually, for water, we use generally an optimal, sort of a modified SBC potential, because that's been shown to be the best fit to the neutron data. But basically, we first create, let's say, a reference starting model. And on this starting model that you work with is you run something called a Monte Carlo simulation on it. And basically what you're doing is you're randomly sampling the box to see what it's in there to gain a total structure factor fit for it. Effectively, what you get is the data you'd expect if you did a neutron diffraction experiment on the box. Now, what we then get EPSR to do is to introduce an empirical potential. And the idea of this empirical potential is it's meant to be the difference in the energy, in the potential between our starting model and the actual final data. The chances are our starting model will not be exactly right, and this is why we introduce the empirical potential to drive it to fit the data. So for example, we can look at this process here. So if we were to zoom in on some of these, we can see once we've implied an empirical potential, if we compare these two, we can see we have a better fit to the data. So we introduce this to drive, say, the movements and so on, so as we get a better fit. Once we have obtained a good fit to the data, we accumulate numerous iterations of the box, so we obtain enough statistics. And from that, we can use that model to extract that atomic level information from it we need. Now, there are a few different things I'm going to use to sort of present data to you, and I will just go briefly through these now. The first one, of course, is the rate of distribution function, where every peak is structured due to bonding with respect to distance. Now, basically, let's say we have a peak here, this represents a kind of prevalent bond that we have in a system. And the first peak that we have here is something I'll refer to as the first coordination shell. Now, quite often what you can do is you can integrate an area such as the first coordination shell. And if you integrate a given area with a given distance, that will give you something called the coordination number. And pretty much for a given specified distance range, that's just going to tell you the number of atoms in it. Now, the other thing that we do also is we can create these spatial density maps. So if you think of almost like the radial distribution function as like a 2D version of showing the location of molecules with respect to distance, you could almost think the spatial density map as a 3D version, where basically what it represents, the density represents the location of surrounding atoms around a central group, basically, a bit like this. Now, we actually create our maps by sending our data through another program called Angular to create these particular pictures. And the color of the density that you can see here represents the density of atoms per angstrom cube. So it's related to that coordination number value. Now, I will show you these in two forms. We can get a map that can either be shown as a distance range measurement, so such as showing the number of particles over the coordination shell, or another one I might show you as well is something called a nearest neighbor, which instead of trying to go within a distance range, just only focuses on getting the location of particles of the very closest particles that are present. And I'm going to tell you which one, and I will remind you what goes on. Now, as I've said before, what we've tended to do when it comes to neutron diffraction is we've generally started small and worked our way up in increasing complexity. And what this does is this models basically the process we see in a lot of, say, synthetic biology systems where we have the bottom-up approach. Start small to look at something big. What this means in our system is even if we start looking at a smaller molecule, we can use this, do these studies, and still extract information on the hydrophobic, hydrophilic, and aqueous interactions, if it's a water system, to try identify key things that could be important for if we want to study a more complex system. Now, to describe this process, I'm going to start by focusing on peptides and amino acids. These are some of the first ones that tend to be studied with this technique. So, as I say, amino acids, of course, are the building blocks of peptides and proteins in our body. And what I have here on this screen is one called L-Glutamine, which is one of those 20. And it's one I actually focused on during my PhD. So L-Glutamine has some important relevance in the realm of disease, because if you get sequences of glutamine, basically, this is associated with neurodegenerative diseases such as Huntington's, where a mutation can happen and the chains can extend, and you can form plaques that you see in disease that cause bad things. And what we wanted to do when we first studied this was just understand how this molecule was hydrated and how it interacted to possibly form these sorts of structures. So what I have on the screen, we have the molecule in the center, and what I have here are radial distribution functions showing the interaction of water oxygens around each of the sort of the polar groups as molecules, the nitrogens and the oxygens around here. As we said, this is the first peak is the first coordination shell, and if you integrate the error, you will get the coordination number for each of these, which is these numbers shown here. Now, if you compare these radial distribution functions, you can see where we have the backbone at the bottom, which is common to all amino acids, and the side chain, which defines what is the L-glutamine molecule. You can see for the backbone interactions, these peaks are higher, these coordination numbers are higher. So they're generally, these groups are more hydrated than these ones at the top on the side chain. We, of course, don't just have to look at hydration interactions. We can also look at, say, glutamine interactions, which is what was important here. One important one was looking at, say, the interaction of the side chain oxygen with the backbone nitrogen. It's a slightly different molecule, but something thought to be common in these sorts of structures. But in this case, our studies didn't show very much. But it means that with these studies, you can explore the hydration association interactions in these systems. Now, if you can gain this kind of atomic level information, what it's then possible to do is to extend this to see if we make a small change, how can we correlate the hydration, the changes in hydration or interactions with the biomolecular properties. So on the left on this screen, I have L-glutamine again, which I'm going to show in red, and a molecule on the right here called the glutamine peptoid monomer. And the difference between these two molecules is simply I have moved the side chain group from being on this central carbon to now being on this nitrogen. And believe it or not, by making that small change, this actually increases the solubility of this molecule by nearly 10 times. Now, these peptoid monomers are sort of the building blocks of a class of peptidomimetic called peptoids. Again, they're very similar in structure, but by making this change, it improves their properties. So they're more robust, they're more flexible, and they're being used as new materials. Now, the question is why, if we've made such a small change, are we seeing such a difference in solubility? And we can use these sort of radial distribution functions again to answer this. So if we take one here, what we have here is the interactions of the backbone nitrogens with water oxygens. And what we can see with glutamine in red again and the peptoid monomer in blue is that the blue peak is smaller than the red peak. And that makes sense. There is one less hydrogen here, so there's fewer interactions that are going to be taking place. But what is interesting is if we actually go and look at the side in this case, let's say at this sort of methylene bridge group over here. Because what we do is if we actually look at this group, which is an area that's not really thought to hydrogen bond well, what we can actually see in this case, which is shown by these groups, is the fact that for the glutamine peptoid monomer, this interaction does seem to have actually increased. We seem to do have an increase in some of the weak hydrogen bonding that's going around. Now, for this molecule, the answer to the solubility question is simply the fact that by looking at hydration, what seems to be happening is even if some of these groups have decreased in hydration, more parts of the molecule seem to be forming hydrogen, even if weak hydrogen bonds with water, and this could account for increased solubility. So it's possible that we can correlate hydration to certain biomolecular properties. Now we can increase this. We don't just have to look at amino acids, and we can increase this to looking at something like a small peptide sequence. So we now have a sequence here of three amino acids, glycine, proline, glycine. And again, we can probe the hydration of different functional groups in the system. So we have like the hydration around the glycine oxygen 1 and number 3. And you can see that for glycine 3, it seems to be higher, the peaks are bigger. And if we actually look at these distance range spatial density maps, we have a broader density going on here compared to this one, suggesting it's more hydrated. Now, when we come and look at these sorts of systems, what we don't just notice is we can understand the hydration of them, but we also noticed in this particular system, the fact that we can actually see water molecules that bridge between two different groups, which is shown by this peak here, by assessing the probability of forming direct hydrogen bonds between the first versus having just water in between. So this suggests that actually having water molecules, not only can the molecule be solvated, but the water could be actually having an active role in the biomolecular function and folding of these structures. Now, is this something that we've seen somewhere else? Yes, it is. We see it in other things such as pharmaceutical molecules, such as prilocaine and cocaine. So if we take cocaine, for example, cocaine is one that can pass through the blood-brain barrier. And what has to happen for it to do that? It has to kind of shield its hydrophilic groups to be able to pass through. And it's thought that that might be actually possible by having a bridging water present. So again, this is suggesting the fact that water could play an active role in the sort of function of molecules, and that we can detect this. Now, finally, we don't just have to look at small thremers. We can go up to longer sequences, such as, say, a helix, such as what we see here. And as we increase our neutron studies, it's still possible that we can actually start looking at not just general hydration, but still look at hydration at specific locations. So if we take this radio distribution function here, this is showing the interactions of water oxygens with the backbone amide group, which is where we're seeing direct interactions, which is happening before, in this case, unfolding after. So what I want to do is take a breather just for a second, just so you can digest that, because I've given you quite a lot of information. But if you were to take anything from what I've just said so far, it's simply as follows. 1, it's favorable to explore the atomic level structure of biological molecules in the solution state, because it allows us to sort of explore both the impact of the solvent as well as the biomolecule itself. Now, to do this, we can turn to other multiple techniques, but we could also use neutron diffraction, because that gives us direct atomic level information in solution and using some kind of experimental probe in that. And so far, when we just start looking at these systems and studying them in water, we can see that water might be playing an active role in the function of biological molecules. So as well as just exploring how hydration influences biomolecular properties, we can see it taking an active role, such as the presence of bridging waters, which could be involved in the formation of, say, certain turn structures, such as the beta turns we saw for those thremers. Now, I've so far really just talked about water. Now, you might be scratching your head going, Tasha, not everything's just water, and you'd be correct. So if we were to take a step back and just think of a biological system such as the cell. So if we were to zoom in on the cell and say the cytoplasm in the cell, you wouldn't just see a water, you would see a mass of different ions, molecules, everything. It is a crowded solution. So realistically, when we want to study a biological system, we don't just want to think about increasing the complexity of the biomolecule. We also want to think about increasing the complexity of the actual solvent, including more and more components, so as we can at least aspire to be something a bit more akin to what we might see in vivo. So what we're going to do is I'm going to now introduce you to sort of a full study that kind of looks at the solvation of lipid structures in something that is not only just water, to explain this. So now we're going to change to looking at lipid structures. So lipids, of course, are a very important part of our bodies, and they form the membrane structures that we find in the cells and everywhere. Now, lipids can also be used in different applications to form different structures, such as the ones that you see here. Now, what determines the structures that are formed is dependent, of course, the actual atomic level structure of the lipids themselves, but also the actual solvent the lipid interacts with. We know fully well, biomolecular studies, the solvent the molecule is in alters its properties and the way it behaves. But of course, again, what we are still lacking is that atomic level information on the role of the solvent on the formation of these different structures. And this is where we want to bring in neutron diffraction, because we can use it to observe the most likely locations of, say, water and of other solvents and how they will bind or interact with of these different structures, so as we can understand this assembly process better. Now, this is not only just relevant for biological systems, this is actually also relevant for areas such as the pharmaceutical industry. So this is what I want to introduce to the sort of realm of things such as co-solvents that you find. So in the pharmaceutical industry, water is generally the most used solvent. But what often happens as well in different mixtures, let's say take from your Voltarol or different creams, you will have co-solvents that are added in to sort of optimize and improve the properties. For something like, say, your Voltarol here, where we have the molecule, we're going to focus on propylene glycol. Something like adding propylene glycol will actually help improve the apparent solubility of the molecule so it can permeate through your skin. Now, propylene glycol is something that's found in a lot of different products. Actually, it's a very common component of antifreeze and actually is a preservative in food. But another area, let's say in the pharmaceutical sector, is actually using it to develop and produce drug delivery vehicles, such as these liposomes. So you'll have these sort of liposome structures where a drug will be encapsulated, so as this whole structure can be carried to its target destination, and it can help it carry there and be soluble. But again, for forming these liposome structures, the question is, what is the benefit of adding a co-solvent here? So something like propylene glycol is usually added because it helps the structures be more permeable and also help it be more, help the drug actually dissolve in that aqueous space in the center. But the question is actually, why is that the case? Now, for areas such as the permeability of the molecule, this is thought to be related to how the propylene glycol would interact, particularly with the head group of the lipids we do. Now, a common lipid that we would use to study to form liposomes would be these type called phosphocholine lipids. And so if we want to ascertain how they can affect permeability, we want to know the information on those atomic interactions it forms to explain why. So in this particular neutron study, what we did is we took a model phosphocholine lipid that is mostly a head group to understand how it interacts with the different portions of the head group, to understand why it would have this impact on permeability, as well as understand the sort of solvent structure so we can understand how it could improve the solubility of the drug. So let me go and walk you through sort of the results that we found for this. So what I have on the top panel are spatial density functions showing the location of water oxygens around the different three main functional portions of the head group. So the choline group, the phosphate group, and the ester group. This is the location of water molecules around, and this is a nearest neighbor spatial density map. Now, what you can see is because if there is density around, this suggests that water molecules are present around these groups. Now, in these two left ones, what we have is a very defined shell. And what that represents is the fact that there is good coordination of water around because it's creating a very defined pattern. This is compared to our ester group, where it's a bit of a diffuse mess. So there's probably not as much coordination of water around this group. We can visualize this or quantify this using those coordination numbers we said about earlier. So by quantifying the amount that would be in the first coordination shell for something like this choline group, right at the top of the head group, we can see that there would be 10, somewhere around 10 water oxygens that surround this group compared to the ester, which is about 2. Now, what we can also do is we compare this to previous studies, which are before we've added propylene glycol and after. And what we can see is that if we do this, that we do see a decrease in water molecules that surround these groups when we do have propylene glycol, but it makes sort of sense. There is an extra solvent in there competing for bond. Now, what I want to do, keeping those spatial density maps for the water, is I now want to introduce comparable spatial density maps for the interactions of propylene glycol. So in this case, I've selected one of the hydroxyl groups to focus on. So it's the location of the hydroxyl oxygens around the different groups. And compared to our spatial density maps at the top four, we've had very defined shells. All of these pretty much look like a diffuse mess. And what this suggests is the fact that propylene glycol, though it is present, it interacts quite weakly with the head group. Again, this is something we can quantify, again, with those coordination numbers, where let's say we take the choline nitrogen group, where we had 10 atoms, we now have a mere 1 for the choline nitrogen. And this amount doesn't really improve, or it actually gets a bit worse when we go through our different functional groups, the phosphate and the ester group as well. So what it already shows is the fact that we're able to ascertain how different solvents in a mixed solution interact with the different portions of a biomolecule. And in this case, propylene glycol doesn't interact very well. Now, this is going to be a slightly difficult one for me to go through, but I ask your patience. And basically what we're going to do is now think about what goes on when we look at generally the solvent structure. So going away from just focusing on the lipid solvent interactions and just thinking about what is the impact on the solvent structure in general when we have a lipid and don't. And what I'm doing here is I'm going to compare the interactions of propylene glycol versus Dmso. Now, Dmso could be used as a solvent, but the problem is it's very highly toxic. And the reason is for this can actually be shown in radiodistribution functions. So what I have on the right here are radial distribution functions showing the solvent interactions for water-water oxygens between the two, and the interaction between the Dmso and water. Now, what happens is that when you actually add the lipid in, what we see is a drastic increase in the number of Dmso water interactions. And what we think is actually happening in this case is the fact that when the lipid gets added, it is dehydrating the lipid molecule. And this could have a notable effect on things such as the permeability and whether that's really good for your sample in general. If we were to compare it to propylene glycol, if you were to add a lipid structure, which again is shown in red versus just the pure solvent, which is in black, we don't really see this. Even when the lipid is in there, the solvent structure is very much maintained. Now, what we think is when we've studied propylene glycol previously in these sort of conditions, we found that it actually quite readily mixed. And so what we can see here is the fact that propylene glycol doesn't cause too much perturbing effect, which means it's hopefully not going to be too toxic to a biological system. And for the fact it mixes, it's very likely its mode of action, if actually increasing solubility, is by it just being mixed in the solution, basically. So those conclusions of that is pretty much that propylene glycol will interact weakly with the head group with a slight preference for proline interactions. Now, what this means for the permeability is the fact that one reason it could be more permeable because it has no distinct preference to interact with a specific group, maybe slightly with proline, but not enough, and could mean it could more easily pass through the liposome structure. And what we also saw is the solvent structure was maintained, which means that chances are the solubility, increase in solubility, is the fact that everything can readily mix in solution. So we'll take a pause again and we'll go through the different conclusions again. I'm going to add an extra one in here. So again, what we saw from before is that it's favorable to explore the atomic level structure of a biological molecules in solution, and we can use neutron diffraction to do this. Now, many studies have been done on water where we have been able to see that water can play an active role in the function of biological systems. But we can move on from just only looking at water, and then it's possible to explore the role of different solvents and solutes for a biological system as well, or even a more biomaterial or pharmaceutical system. Because what this does is increases our understanding of looking at more complex systems, and it means that we can actually start doing studies like what we've just seen here for C3PC and propylene glycol for different applied settings, such as, say, the pharmaceutical industry, for instance. Now, what I'm going to do to sort of end this, we're going to go less into going very specific into studies and just more take a bit of a perspective. Let's think about this, thinking about where neutrons are having impact and maybe what their future is. Where do they play a role in being integrated into this realm of sort of biophysical techniques? So let's think about impact first. So I showed you earlier the slide showing the different molecules that have or are being studied. But what we're actually seeing is that not only are we doing these fundamental studies, they are having impact. So we've got about 3 here that we can do as an example. So for example, if we go back to one where we discussed cocaine, what we've been doing is we've been studying sort of different drug molecules in different solvent conditions. And by understanding how they interact with different kinds of solvents, sort of hydrophobic, hydrophilic, we can correlate those properties to understanding how it might permeate barriers such as the blood-brain barrier. If we understand interactions like that, we can possibly optimize the development of new drugs. Another one that we completed is, say, things like studying the hydration of sugars. Now, we don't have very much information on receptors that sugars can bind to on your tongue. So the idea of doing such studies is we could use neutrons to do hydration studies to see if there's any correlation between hydration and the sweetness. Now, in this case, we actually found there was a correlation between those factors. So hydrogen bond length and those hydration interactions were correlated with sweetness, which might mean that we can use such hydration studies to do simple studies on such sugar solution systems to understand sugar interactions better, as well as possibly the development of new artificial sweeteners. And finally, a little bit less from thinking about different biological systems, we can think about just simple salts that are relevant to the origins of life. So we've actually studied things such as perchlorate salts and had their interactions with water, because perchlorates are thought to actually allow liquid water to exist on Mars. And if we study these solutions, different things at the right conditions, we've been able to ascertain, using such neutron diffraction studies, the impact those salts are having and why liquid water is actually possible under certain conditions in Mars. So there are different things that are coming out that are having impact and are being picked up by news. I've purposely picked some of the more scientifically relevant ones. Ones like the mimetic Martian water has some very interesting new studies. So if you look that up, that is an entertaining read. But in the end, of course, we have all these impactful studies. And of course, now what we want to do is we want to keep extending using this technique to study more complex systems. So that could be just expanding the repertoire of molecules we study, starting to look at more complex assemblies or materials, or simply just actually increasing the size of the molecule that we're studying. So just say, trying to look at the solvation of a protein, for instance. So here's actually some example data from looking at a protein. Again, we have those atomic level interactions. But if we actually span that range, we also have the information that we can have on the bulk structure on the protein as well. And if the idea is the fact that the solvent structure around there, we might be able to interact, we might be able to extract that information on the solvation or hydration shell of such molecules. This is what we kind of things we hope to do. Now, when it comes to integrate into this sort of realm of biophysical techniques, let's say the aim is the fact that we want to be able to use neutron diffraction in sync with other methods. As I say, if we looking at the Venn diagram earlier, of course, neutron diffraction possibly ticks our box of being able to look at direct atomic level structure in solution with an experimental probe, but we kind of want to, I guess, integrate it properly into the realm of different techniques. And there's two ways that we can explore this, and this is what I'm going to briefly do at the end now, which is thinking about how other methods could support neutron diffraction, and also how neutron diffraction can in return be used to support other methods. So I'm going to do this one first and follow the other. So earlier on, I showed you this slide, which was looking at the interactions of a much longer peptide sequence and saying the fact that we can extract sort of site-to-site information on the hydration, like on specific residue. Now, this was my little clue over here because I didn't really talk about this, but the truth is that this is not just actually a neutron diffraction study. And what we've actually had to do in this case is when we've come to do our analysis, we have actually been using multiple techniques to then feed in a structure that is suitable for our EPSR analysis that we can then gain information from. So this is the actual process that we did. So what we were doing in this particular peptide is we were studying this peptide in methanol water solutions at different concentrations. And the idea of studying this particular peptide was the fact that it forms this particular amphipathic helix where the polar residue sits on one side and the non-polar one sits on the other side. And the idea of studying at different concentrations is the fact that we might be able to ascertain what induces folding, because these are structures that are thought to be involved in the sort of common feature in antimicrobial peptides. So we might be able to understand better what allows them to function as they do to form better ones. Now, what we would need to do in this case is what we've wanted to do is be able to actually gain neutron diffraction studies to gain insight into the atomic interactions at different intervals of the unfolding process. But the thing is, it's actually quite difficult to then put in sort of a starting structure that the EPSR method could work with and optimize. So what have we had to do? First of all, the actual process was we did circular dichroism spectroscopy, which is a probe of secondary structure, to gain an idea of the sort of helicity of the structure at each stage. We then confirmed this doing neutron diffraction, NMR studies, which look also at the conformation. And because the solvent environment, the chemical shift are sensitive to solvent environment, we do gain some information on the solvation as well. Now, with an understanding of the sort of these experimental probes that give us information on the conformation, what we've done then is we have completed molecular dynamics simulation. So molecular dynamics is a sort of measures the time evolution of a system, basically. And what it does is once you've set your parameters, it basically runs a simulation by resolving Newton's laws of motion. And when you've accrued enough time steps by sort of resolving velocities and positions at each stage, you gain enough information and can gain these sorts of structures. So what we have done is we have run an extensive molecular dynamics simulation, which with a starting structure has basically trended towards going towards an optimal conformation for each of these solution conditions. What we have then done is using that and comparing them against that, which we've found a correlation between the two, we have fed those structures into our EPSR analysis. So what we've actually done in terms of other methods to support neutron diffraction is what we can do is we can use biophysical techniques to introduce a suitable reference potential in the first place for the analysis that we need to do. Now, the other way, thinking about how neutron diffraction supports other methods. If we go back to the beginning, pretty much we already know the fact that neutron diffraction can be used to have this atomic level resolution on a system where you can gain information, the conformation, bond interactions, the bulk solvent structure. And even if we, depending how complex we can go, even if we can do this on even more simpler systems to inform more complex ones, we still have that unique insight that many other methods don't have. Now, what's really handy is that actually the instrumentation for neutron diffraction and other techniques has actually improved. So if we were to actually compare sort of the distance ranges that can now be covered, let's say with one of the more recent instruments, we can actually see for neutron diffraction down here that it actually overlaps well with small angle neutron scattering, which is a technique that looks at bulk structure. And what that means, therefore, for a given system, if we were to use that, as well as another technique like that, we can transcend the whole level from just looking at atomic level structure to looking at bulk structure as well. But finally, what neutron diffraction could be useful to do. So we've talked about feeding in information from other potential, from other techniques to give a sort of starting model. But what we could actually do in return is use neutron diffraction to help us develop that as well. So what we actually have, for example, is when we actually sort of set up our simulation, we actually create a model, use force fields from the literature and see how well they fit. And there are certain biomolecular systems that are more complex or where you have interesting charge systems where established force fields don't really exist or they're not very good. So this is where we can actually go beyond and we can introduce things as density functional theory, these quantum methods that can be done. But nothing is stopping you from trying out different potentials, such as those from density functional theory, and actually comparing them and using the data as a probe to confirm how good the potentials are. So what we can also do in return is use neutron diffraction as a means to support force field development for computer simulation as well. So overall, start wrapping up and ending. So again, we can use neutron diffraction to study the direct atomic level interactions of biomolecules in solution. And these studies have been used to show that we can actually extract this information for both systems containing water and those that are in mixed solvents. What we can also do, though, in terms of the long-term goals of neutron diffraction, is what we're hoping is that we can integrate this more as a core technique in sort of the biophysical realm of understanding biological structure. And what we can do is both integrate it, bring in other techniques to help support our neutron diffraction analyses, but use neutron diffraction as well to say, bring in that atomic level insight that other methods don't do, and also use it to help us develop new force field parameters for areas such as, say, the optimization of computer simulations. And those are the conclusions that I have. So finally, what I need to do is I need to thank a lot of people who have been with me for the past 11 years. So one of them is the group I'm affiliated with, so the Lorenz group at King's. And I need to thank all of my colleagues, collaborators, my students that I've worked with and so on for all of their help over the years. Finally, I guess I need to also thank the SDFC for Neutron Beam Time, all these people for funding here, King's, of course, for funding me currently, and really to thank you for your attention. I mean, we will have questions now, but you'll also be able to contact me here. Thank you very much.
Thank you. It was an amazing talk. It was very impressive how, your work has applications from pharmaceuticals even to water on Mars.
Thank you.
It's very impressive. So now, if anyone has any questions, you're very welcome to unmute yourselves or raise a hand or type in the chat. I'll give a minute for everyone to gather their thoughts.
They've had a lot of information. So.
In the meantime, I was wondering, in terms of the complexity of the solvent system, what is the next level you're aiming to to achieve in terms of testing the interactions and so on?
That's a very good question. I mean, I guess for a biological system, the very obvious one is to just ensure that we're using solvents that pretty much you use maybe in some in vitro studies. So quite often we haven't really done much studies in, say, something like PBS, which is actually a very solvent, which of course is a mixed, ion solvent system, which is used to do the pH. That would be a very obvious one. But what we can keep doing, as I say as well, is we can keep adding gradually bit by bit, adding a different component. So for example, there have been studies that have been done of cryoprotected solutions, which I think was about a 4-component solution study to see why that is the case. So I think in the cases, it's just building up and up. And I'd say which ones it is going to be very dependent on the application that you have. But there's lots of ways you can go. So,
yeah. Well, that makes it even more exciting. We have thank yous in the comments in the chat. Does anyone have a question? So far, no one. Oh, Karen.
I have a question, if that's okay, Tina.
Yeah.
I'll just turn on my video quickly. There we go. Thank you, Natasha, for such a fascinating talk. I really enjoyed it. It's really fascinating how you can start to shed some light on some really complicated questions. And it really struck me that in my field of chemistry, we see researchers all the time kind of thinking that choices they make along the way might not be consequential in terms of the activity they try and see. So they might be spiking a molecule into a biological system, for example, in an organic solvent, varying the concentration of that organic solvent, and just assuming that because there's so much water around, that other solvents are going to be doing anything. And I think it's really fascinating that your work can really show that that might not always be the case. Is that something you find quite a lot, that researchers have a tendency to assume things aren't going to have a big effect on the function of the molecules they're testing, whereas actually it's always a lot more complicated than we expect?
That's a good question. I think it's very mixed in many cases, because I think there are some biological studies where it's just like, we've just done this system, we haven't really thought about the solvent, and it's a bit of a mixed bag. So I think there could be some studies that do. There are some studies where we get like, be fine, it's just the standard that we use. And possibly maybe what it is more is the fact that there may be certain conditions that we sort of assume that we just use anyway, and it's maybe knowing what actually the effect that it has, and maybe that's not 100% clear, and maybe this is where some of these studies could feed in. So
yeah, thank you. Really, really interesting stuff.
You're welcome.
Thank you. Can I ask one more question? We have a comment in the chat. Thanks for the very interesting presentation. And I'm very interested in the brain-blood barrier part. So yeah, I was wondering in terms of, because everything is connected to biology, physics, chemistry, do you have to collaborate with, I mean, there's a lot of people, I assume that you have to collaborate, but in terms of someone that is completely not related to neutron diffraction, or is everyone sort of, I mean, like, is it everyone connected to the neutron diffraction theme, or just have to have a broad collaboration with other scientists?
Let me check I've got your question right. So is it the case of is the neutron diffraction a sort of a niche area? Is that what is that?
Oh, like, because you have to consider both like, for example, in the biological, like pharmaceuticals, you have to have a specific person that is a specialist on pharmaceuticals, for example.
That's a good question. I mean, I guess I think with some of the neutron stuff, I guess it's been done by quite a niche niche audience, sort of a niche collection. I guess what we've had to do is slightly reach out to people who are a bit more expert in those areas. So there are people who have a specific interest in that sort of stuff. But yes, so I guess, yes, I think it's sort of a growing thing. I think what's happening with time is it's an area where fewer researchers have done, but it's starting to be found. And so we've had to start reaching out to people, and they in return are starting to come there, and it's increasing its repertoire because we've had, again, you have to start quite small. Because we've had to start small, basically, what it means is the fact that we can't, what am I trying to say? Because we've had to start with smaller molecules. It's something that maybe hasn't been caught the attention of maybe the biological audience is the same. Can say, oh, why do I want to study an amino acid or something like this? But I think now we've got that mix coming in where we're overlapping length scales. Now people go, hang on. So this could be cool now. So yeah, this is where we're starting to see it. Have I answered your question?
Yeah, I think so. Yeah, because you're expanding now. Yeah, it's very exciting. I think it would be very useful because as I'm doing medicinal chemistry, especially in the pharmaceutical parts, you have to consider like everything because the interactions are quite complex.
Yeah.
Of the drug with the target you want to achieve, and it's
no, exactly.
It's very useful.
Very true.
Can I ask a last question?
She can.
I want to say thank you for an incredible talk. It was really interesting. I was wondering, just personally, what is the most exciting part of your job? What makes you get up every day?
Oh, that's a good question. That is a very good question. I guess over these, what's going to be my favorite? I think I do a lot of different systems, so I guess it's bonus. I'll tell you what it is actually really very recently, because I guess as I've actually gone along, I guess I've progressed through doing different studies and different so on. But actually the more recent one actually has been them being able to pass this knowledge on to the future generation and do things like this. And so, for example, like say my student here, Felix, has been working on some of this stuff, and actually seeing them flourish and actually work in this as well. Because in some ways, especially given this is quite a niche area, niche area that's sort of expanding, it's kind of nice to see things grow. I mean, there are, in the repertoire, there are some very cool studies that say about like Mars and things like this. And knowing you're doing a purpose, like some of the material stuff, I get driven. But I'll be honest with you, actually, that aspect of passing it on and actually watching the students flourish as well, actually, that's my thing. So,
that's really cool. Thank you.
You're very welcome.
Yeah, thank you again. I'm gonna stop the recording now, but everyone is welcome to stay for a bit more informal chat.